All right, lecture 4 for chapter 7, the electron transport chain and oxidative phosphorylation. Oxidative phosphorylation is the second of the 3 types of ATP synthesis that I promised you back in lecture 1 we were going to talk about. So the first type was substrate level phosphorylation. The second one is oxidative phosphorylation. And oxidative phosphorylation is basically using the energy that is pulled out through redox reactions in the presence of oxygen in order to synthesize ATP. Okay? So, well, there we go. Using the energy from redox reactions in the presence of oxygen to synthesize ATP. Okay, now the electron transport chain and oxidative phosphorylation take place in the mitochondria. In particular, the electron transport chain, not sure why I'm having trouble writing here. The electron transport chain takes place across the inner membrane of the mitochondria. And then ATP synthesis is done by an enzyme in the inner membrane. Okay, other important locations are going to be the matrix and the intermembrane space. So all that mitochondria anatomy stuff we talked about last lecture, you're going to need it in this one. Okay, now, the electron transport chain is essentially a series of redox reactions where electrons obviously are transported. And remember, every time we move electrons around, we're moving energy around. But this time, that energy is being used to do something else. It's not just being transported to an electron transporter and then moved somewhere else. We're actually doing something with it. Okay, so let's orient ourselves a little bit in this figure. Okay, so the light yellow part is the mitochondria matrix. This is the phospholipid bilayer is the inner membrane, and then this orange part up here is the intermembrane space. Now, these big purple blobs, those are the transmembrane proteins that make up the electron transport chain. And there are 4 of them. They all have unique names. You don't have to worry about that. Okay. So what happens is that NADH, which is present in the matrix of the mitochondria, either from being generated in the citric acid cycle or being generated in glycolysis and diffusing into the mitochondria. Regardless, the NADH is in the matrix. And NADH is going to give its electrons to complex 1 in the electron transport chain. Anytime electrons are transferred, you're going to have a redox pair. Okay, so if NADH is giving away its electrons, it's losing them. So NADH is oxidized back to NAD+. complex 1 is reduced because it gains those electrons. Okay? Well, complex 1 is going to use some of the energy in those electrons to actively transport hydrogen ions from the matrix into the intermembrane space. Okay, now remember, active transport, moving things from low concentration to high concentration. So we're going to have low hydrogen ion concentration in the matrix. We're going to have high hydrogen ion concentration in the intermembrane space. So we are creating a concentration gradient across the inner membrane. Okay? So NADH transfers its electrons to complex 1. Complex 1 uses some of the energy to actively transport hydrogens. Then those electrons are picked up by an electron shuttle. That's what this little Q molecule is. And those electrons are transferred to complex 3. Complex 3 uses more of their energy to actively transport more hydrogens into the intermembrane space. The electrons are transferred one more time to complex 4. Complex 4 actively transports more hydrogen ions. And then those electrons have reached the end of their journey. Well, what's waiting on them at the end of their journey? Oxygen. So those electrons combine with hydrogen and oxygen to form water. Okay? Well, what about this FADH2 in complex number 2? Remember I told you in lecture 3 that FADH2 is an electron transport molecule. It's like the first cousin to NADH, but it is not quite as high energy a molecule as NADH2. So the electrons that FADH are carrying, they don't have enough energy to go all the way through the electron transport chain and drive the active transport of hydrogen 3 times. They only have enough energy to enter the electron transport chain a little bit later at complex 2 and drive the active transport of hydrogen only twice. Okay? All right. So at the end of the electron transport chain, we have generated a hydrogen ion gradient. Okay? Remember that ion gradients are sources of potential energy. Okay? So we have, if this is the inner membrane of the mitochondria, and this is the matrix down here, where we have low hydrogen ion concentration, and this is the intermembrane space up here, where we have high hydrogen ion concentration, we have a concentration gradient. It is a source of potential energy. Well, these hydrogen ions naturally want to move from high concentration to low concentration. Why can't they just diffuse across the inner membrane? Because they're polar. Okay, remember, the inner membrane is a phospholipid bilayer. It has a hydrophobic interior. Those hydrogen ions can't just go back through the phospholipid bilayer through simple diffusion. What do they have to have? They have to have a transport protein in order to do that. That's what ATP synthase is, okay? ATP synthase is this massive enzyme complex that couples the diffusion of hydrogen to ATP synthesis. So remember, when hydrogens diffuse down their gradient, energy is being released. It's like sliding down the slide at the playground, okay? So when hydrogens diffuse through ATP synthase from high concentration to low concentration, energy is released. ATP synthase then uses that energy to take ADP plus phosphate and synthesize ATP. Remember, ATP synthesis is a highly endergonic reaction. Okay, we've got a big positive delta G here. We've got a big energy of activation. Where does that energy come from? That energy comes from the diffusion of hydrogen ions down their gradient. And that enzyme couples, remember it links those two processes together. The diffusion of hydrogen ions that releases energy to the synthesis of ATP that requires energy. So ADP plus phosphate is used to make ATP. Okay, so this is the oxidative phosphorylation reaction. But the energy that's required for this reaction is provided through all of those redox reactions where electrons were transported, and then the energy in those electrons was used to actively transport hydrogen. The hydrogen ion gradient was then the energy source for ATP synthesis. So if we put all of this together, all of aerobic respiration together, remember our end goal was making ATP. Glycolysis, pyruvate oxidation, and the citric acid cycle, their purpose was to break the chemical bonds in food molecules and their intermediates to pull energy out. Most of that energy was in the form of these electron transport molecules, NADH and FADH2. The electron transport chain further harvests the energy from those electrons to create the hydrogen ion gradient. And then the energy in that gradient is used to generate ATP by ATP synthase in the inner membrane. Okay? Chemiosmosis is another word that you're going to see when we're talking about aerobic respiration. I don't tend to use it a whole lot, but chemiosmosis is referring to the process of using the energy in the gradient to generate ATP. Okay? Now, I know that that's a lot, and you're going to have to just spend a little time wrapping your head around how electrons correlate to active transport of hydrogen and how that correlates to the energy to make ATP. Okay, you're going to have to spend some time drawing some diagrams and just practicing that information. I'm also going to post the link to some animations that may help you visualize